If we take a cross-section of the kidney, there are two main parts, the outer cortex and the inner medulla. If we zoom in, there are millions of tiny tubes called nephrons, which go from the outer cortex down into the medulla and back out into the cortex again. Nephrons filter out harmful substances in the blood so that we can excrete them into the urine. Each nephron is made up of the glomerulus, or a tiny clump of capillaries, where blood filtration begins. These capillaries have very thin walls and they act a bit like a coffee filter. Red blood cells and proteins that are large stay in the capillaries, whereas blood plasma and smaller particles get filtered out. This filtrate, called tubular fluid, collects in a cup-shaped structure containing the glomerulus called the Bowman's capsule. Together, the glomerulus and the Bowman's capsule make the renal corpuscle. The Bowman's capsule is connected to the renal tubule, which has a few segments. First, there's the proximal convoluted tubule. Then there's the U-shaped loop of Henle, with a thin descending limb, a thin ascending limb, and a thick ascending limb. And finally, there's the distal convoluted tubule, which empties into the collecting duct, which collects the urine. Zooming in on the distal convoluted tubule, it's lined by tubule cells, which are similar to the ones found in the proximal tubule, but they don't have microvilli. On one side is the apical surface, which faces the tubular lumen, and on the other side is the basolateral surface, which faces the interstitium, or the space between the tubule and the peritubular capillaries. The peritubular capillaries run alongside the nephron and return solutes and water that were reabsorbed into the interstitium back into the circulation. The distal convoluted tubule is split up functionally into the early distal convoluted tubule and the late distal convoluted tubule, which is very similar to the collecting ducts. The early distal convoluted tubule is impermeable to water, and the tubular fluid contains more sodium than the tubule cells, so sodium ends up flowing down its concentration gradient into the tubule cells, using various protein channels. Some of these channels are co-transporters, meaning that they move two or more different solutes at a time. One example is the sodium chloride cotransporter on the apical surface of the early distal convoluted tubule, which moves one sodium and one chloride ion into the cell. The cotransporter moves sodium in the direction of its concentration gradient and uses that energy to move a chloride ion against its concentration gradient, meaning that there's a higher concentration of chloride in the cell as compared to the lumen. Once inside, chloride leaves via the chloride channels on the basolateral surface of the interstitium, down its concentration gradient. Similarly, there are also calcium channels on the apical surface and sodium-calcium channels on the basolateral surface of the tubule cells. Calcium ions can diffuse across the apical surface into the tubule cells, down their concentration gradient, and then it can move across the basolateral surface into the interstitium against its concentration gradient, as one sodium moves from the interstitium into the tubule cells. Calcium reabsorption is regulated by parathyroid hormone, which is secreted by the parathyroid glands in the neck. When parathyroid hormone binds to the tubule cells, they start making more sodium-calcium channels, which increases the reabsorption of calcium. But before sodium can build up in the cell, it's pumped out through the basolateral side into the interstitium by a protein pump called the sodium-potassium ATPase. The sodium-potassium ATPase pumps 3 sodiums out of the cell and lets 2 potassium ions into the cell from the interstitium. Since there's more sodium in the interstitium and more potassium in the cell, it goes against both of their concentration gradients, and that's why the pump requires ATP. So the sodium-potassium ATPase on the basolateral surface keeps the intracellular sodium levels low, and that allows more sodium to flow into the tubule down its concentration gradient on the basolateral surface through the sodium-calcium channels, and on the apical surface through the sodium chloride cotransporter. Normally, the early distal convoluted tubule reabsorbs around 5% of the filtered sodium, but this reabsorption is load dependent, which means that when more sodium makes it past the proximal convoluted tubule, more is reabsorbed in the early distal convoluted tubule. This is possible because more sodium delivered to the distal convoluted tubule means a stronger sodium gradient for the sodium chloride cotransporter. Now, in the late distal convoluted tubule and collecting duct, there are principal cells and alpha intercalated cells dispersed among the tubule cells. The principal cell has 2 channels on the apical surface, an epithelial sodium channel called ENAC that allows sodium into the cell, and a potassium channel that allows potassium into the lumen. The flow of positively charged sodium ions into the cell helps drive the positively charged potassium ions out of the cell. There's also a sodium-potassium ATPase pump on the basolateral surface that again moves 2 potassium ions in for every 3 sodium ions out. Now, the alpha intercalated cells mainly get rid of hydrogen ions by pumping them across the apical surface into the lumen of the nephron. First, they have hydrogen ATPase, which uses ATP to pump hydrogen into the tubule against its concentration gradient. And second, they have a hydrogen-potassium ATPase, which uses ATP to push 1 hydrogen into the tubule in exchange for 1 potassium, both against their concentration gradients. Finally, it's worth mentioning that, like all cells, sodium and potassium levels are controlled by the sodium-potassium ATPase pumps on the basolateral surface, which move 2 potassium ions into the cell and 3 sodium ions out of the cell. So overall, there's a net movement of sodium and chloride into the blood, while hydrogen is pushed into the tubule. The distal convoluted tubule and collecting duct are hormonally regulated by aldosterone, a steroid hormone made in the zona glomerulosa of the adrenal cortex. In the principal cells, aldosterone diffuses across the basolateral membrane and into the nucleus of the cell, where it triggers the increased synthesis of ENACs, ATP-dependent potassium pumps, and the sodium-potassium ATPase transporter. Together, they work to increase sodium reabsorption and potassium secretion. In the alpha intercalated cells, it increases the synthesis of hydrogen-potassium ATPase to increase hydrogen secretion. Finally, inside the principal cells are water channel proteins called aquaporin II, which usually sit in vesicles inside the cells of the distal convoluted tubule and collecting ducts. When the body wants to retain water, the pituitary gland secretes antidiuretic hormone, or ADH. Antidiuretic hormone binds to the principal cells via a protein receptor, and it triggers the vesicles that have the aquaporin II to bind to the basolateral surface and the apical membranes. This allows water in the lumen of the tubule to rapidly cross the apical membrane and get into the cell, and then continue the journey across the basolateral membrane into the interstitium and nearby peritubular capillaries. This process of osmosis shifts water from the lumen back into the circulation. Alright, so, as a quick recap, each nephron is made up of the glomerulus, a proximal convoluted tubule, a loop of Henle, a distal convoluted tubule, and collecting ducts. The early distal convoluted tubule is able to reabsorb sodium, chloride, and calcium. The collecting duct and late distal convoluted tubule have principal cells, which absorb sodium and secrete potassium, and alpha intercalated cells that secrete hydrogen. ADH can also signal the principal cells to embed aquaporins into the membrane to increase water reabsorption.